Hello. Very good evening to the student who have joined. Can you kindly let me know in the chat box if my voice is audible to you, and also if you can see the PPT. Let me know if everything is fine with my voice and also with the video and PPT. Okay, thank you so much. So yeah, we will wait for till it is 6 p.m. and then we will start today's session. Very good evening to all. Just let's wait for some time and then we will start today's session. Meanwhile, let me know if there is any problem for listening to my voice and for the seeing the PPT. Please let me know in the chat box if you cannot listen to my voice properly, or if you cannot see the PPT. Okay. We will just wait for 2 more minutes and then we will start today's session. Yeah, very good evening to all. Okay, I think I can start with the introduction portion. Meanwhile, when most students will join, I will just add them and we can start this question answer session. Okay. So, very good evening to all. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry, and today it is our week 6 live session. So, till now we have completed 5 live session in last 5 weeks, and today it is our week 6 live session. So, right now the week 7 course content is online, right? So, every week, each and every week we are going ahead with a session, like in first session we had completed week 1 and week 2 both together and so on. So, today we will complete the week 7 course content. Okay, so before going to the week 7 course content and the portion of starting the class, I want to clarify few things. I always before starting the class I just typically repeat these points. So yeah, the points will be like this, that there will be this kind of problem solving session every week. Okay, the timings and this for the session will be from on every Tuesday from 6 to 8 p.m. Okay, and these sessions will be live on Google Meet platform. And if any network problem happen from my side, so please just wait for some time. I will surely rejoin soon. Okay, do not leave the session. Okay, I will rejoin as soon as possible if there is any network issue or power cut issue from my side, and if the problem is from your side, you just can rejoin at any time into the class. Okay, there is no problem, and I am recording the session. Because of that, I have just muted you all and have just kept your cameras off. But if you want to tell me anything, any problem, anything you want to share, just you can write it in the chat box. Okay, the chat box is open always and I am checking the chat box. So if you have any doubt, any query, or you want to tell me anything about the previous sessions also, if there was any doubt at any point, or you want to repeat me anything, just anything you can write it in the chat box. Okay, I will try to repeat that thing, or today or from next day I will just repeat that thing, whatever you have problem faced any problem. Okay, so during the session you can post your question or queries to the chat box, and if still any doubt persists after class, you can drop an email to me. Okay, I will share my email ID at the end of the session. Okay, and if you can just mail me any doubt or your any query. Okay, I will try to answer that as soon as possible. And again, this presentation and class recording will be shared to you soon, so you don't have to worry if there is any problem from the network issue from your side. You don't need to, you don't have the won't be any problem, because I will be sharing this presentation and also this video. Okay, the class presentation video. So just wait for 2 more minutes. I will check if any more students are joining or not. Then we can proceed with our session. Okay, so I think I will just keep this call open so that any student can log in at any time. Yeah. So now it won't be any problem. Okay, so I think I will just start with the question answer session. Okay, so you all know that who have joined I mean who have attended my this live session previously also, you know what we do in every live session, that we cover the 20 around 20 MCQ question related to that week's material. Okay, so we are doing this kind of MCQs because this MCQs you have to you will find similar MCQs in your assignment portion, which is given to you every week, and I am giving you this kind of MCQs which will help you to solve the that week's assignment, as well as I will discuss each and every MCQ problem thoroughly, so that you understand that week's content more better. Okay, and it will help you for your final examination also, and also for overall understanding of your course. But for that happen, I want all of you to answer the questions. Okay, even if your answer is wrong, but I want all of you to participate and answer the question which I am showing to you. Otherwise, I won't understand if you are listening to me or not, or if you are following up other session. Okay, so please try to answer the questions. So today it is the week 6 live session, and since we are going ahead every week, so we will be discussing today the week 7 course contents. Right now the week 7 is online, right? So we will be discussing the week 7 course content. What are in the week 7 course content? The topics are drug administration route, which is divided into part 1 and part 2. Then we will discuss finding a lead, which is again divided into part 1 and part 2, and then drug screening, and we will have a tutorial around regarding drug administration route and lead finding. Okay, so these are the topics which we will discuss in today's course, and the questions all will be based on these topics. So let us start with the first question of today's session. So the first question is, which of the following reflects the order in which various drug discovery and development stages takes place? The options are: number A, target identification, establishing a bioassay, finding a lead, and SAR. SAR you all know is the structure activity relationship studies. Now number B, it says the first establishing a bioassay, then target identification, followed by finding a lead, and then SAR. The third option, number C, it says that target identification followed by establishing a bioassay, then SAR, then finding a lead. And option number D is target identification followed by structure activity relationship or SAR, then establishing a bioassay and finding a lead. So you have to identify that A, B, C or D which reflects the correct order of drug discovery and development process. Like every day I will give you some time, and then I will check the answers in the chat box. Let me know A, B, C or D which of the following is the correct answer, and then we will discuss about the various steps of the drug discovery and development process. So let me now check the chat box. So I have only one answer, which is saying it is number A. Okay, I am getting two answers now. Number A is the given answer. Okay, I will give you 1 or 2 more minutes. Others also try. I want all of you to answer the question. Okay, even if your answer is wrong, again I am saying that it doesn't matter, but answering the question is very important. So which of the following reflects the correct order of various drug discovery and development process? Number A, target identification, establishing a bioassay, finding a lead and SAR. Number B, establishing a bioassay, target identification, finding a lead and SAR. Number C, target identification, establishing a bioassay, SAR, finding a lead. And number D, target identification, SAR, establishing a bioassay and finding a lead. Okay, again I will be checking the chat box. Okay, now I have a few answers. So the answers are number A, number D, number C. Okay, let me check which of the following is the correct answer, and then we will discuss the various stages of drug discovery and development process. So the correct answer will be number A. Target identification is the first one, then establishing a bioassay, after that finding a lead, and then SAR. Okay, now let me show you what is a drug discovery and development process. So I believe in our very first session, I think I have discussed a little bit about the drug discovery and development process and the stages, various stages of the process. So today I will again repeat that part, so that you just remember what we have done in our first week. So this is a flow chart about the drug discovery and development process. So where does it start? The drug discovery and development process. The first is the UMN identification. Now what is a UMN? UMN is a unmet medical need. Okay, unmet medical need. So how this unmet medical need it is we have identified. So first we have to do is the market analysis, and we have to take the input from the KOLs, okay, who are the who know what is going on in the market, the medical practitioners, the doctors and everything. First this kind of analysis we have to perform, and this will give us an unmet medical need, which is the first thing, or we can also say it is the identification of the disease for which we are going to perform that particular drug discovery and development process. Each and every drug discovery and development process is for developing a drug for a particular disease, right? Just like for COVID-19, when there was a vaccine finding was happening, so that particular disease was the unmet medical need, okay. And then we have to do is the target identification, okay. And what is the target identification? Target identification is that any disease, it can be tackled in a various ways. For suppose we have now learned many things about the cancer, right? We have discussed about the cancer, how to treat the cancer. We have discussed few about the anti-cancer drug, or let's say we have discussed many about the anti-viral drugs or antibacterial drugs, and in upcoming weeks we will be discussing about these things in more detail. So what is the way to tackle each and every disease? There are several ways, okay. Just like for cancer, we can just target the malignant cells, the various enzymes like kinase inhibitors are one kind of anti-cancer drugs. There are topoisomerase inhibitors and any kind of inhibitor drugs or any kind of activator drug, agonist, antagonist. There are various proteins which can be targeted, various metabolic pathway, many various your cellular pathway, DNA synthesis pathway or cell division pathways. Any pathway we can target, but for that we then first after getting the unmet medical need or the disease, what we have to do is we have to identify the target. which have to target it for that particular disease. Okay, and how this target identification can happen? We can the phenotypic screening can have be used, genetic association, transgenic organization, imaging, that can be used for target identification. After identifying the target, what we have to do is we have to validate the target. Okay, so the target validation is the third step. So what happened, how this kind of thing can be used? Then we can do the antisense and RNAi technology, genetically modified animals, proteomics. So these can be done for validate the particular target. After target validation, what we have to do is the hit identification. Now hit identification can be done in by lots of way. We can do using the various assays. So so for using the assays, we have to develop or establish a bioassay for that particular target. Then high throughput screening, fragment based screening, or focus screening, which are the basically the in silico portion, okay, which is the hit identification, and followed by the hit confirmation. Hit confirmation also can be done by counter screening, then dose response and functional assays. Okay, after that what happened, we have this lead generation. Now lead generation is the third step, which is given here like this. We can just first the unmet medical need identification, then target identification, followed by target validation, hit identification, hit confirmation, then we have to do is the lead generation. So how the lead generation can be happen? We can do the conventional or combinatorial chemistry, or we can use the SAR develop. Then after lead generation, the what are the various steps is that SAR establishment, like for activity, selectivity, physicochemical properties, ADME and pharmacokinetics, and toxicity potential. Then in vitro PK, primary POM and disease models can be used for the lead generation. After generating the lead, what we have to do is we have to optimize that lead. So what are the steps? So we have the enhancement of physicochemical and ADME properties, then we have PK/PD correlation, metabolic ID and characterization, backup medicinal chemistry strategies. And then after all this step, we will have a particular compound or few compounds which can be used as preclinical candidate, and then clinical trials of that particular drug will start. And clinical trials are also, there are 4 to 5 phases of clinical trials, like phase 1, phase 2, phase 3 and phase 4. Okay, and after the clinical trials, what happened? That the drug is approved for the market surveillance, and then we have a last phase of clinical trial, which is the post market surveillance, which I have discussed in the first lecture itself. You can just go back and check the video on the lecture PPT for that first week's PPT. Okay, now then you will find what is the steps of the clinical studies, and till now we have discussed about the preclinical developments. Okay, so this was the first question. So many of you have identified the correct answer, and others it is fine if you cannot identify the correct answer, but answering the questions are very important. What I have always told you, that please answer the question, even if your answer is wrong, it doesn't matter, but please participate. Okay, so now moving forward, I will go to the next question. So problem number 2 of today's session is, there are several sources and methods of discovering new compounds. Which of the following is most likely to lead the discovery of a complex structure quite unlike any other previously discovered? Your options are: number A, combinatorial chemistry, number B, data-based mining, number C, screening plant extracts, and number D, me-too drugs. Again I will repeat, there are several sources and methods for of discovering new compounds. Which of the following is most likely to lead to the discovery of complex structure quite unlike any other previously discovered? The options are: number A, combinatorial chemistry, number B, database mining, number C, screening plant extracts, and number D, me-too drugs. sorry. So again I am saying, all of you, please try to answer the questions. Even if your answer is wrong, I am not giving any marks for answering the class, but it will help me to understand if you are following my topics or not. Okay, and also if you have understood that week's course content. Let me check the chat box now. Okay, I think I have few answers for this question. Number C, number A, number A, number A, number B, number A. So I have number C, number A, number B. So I have three different answer for the question. Okay, fine, but all of you have answered, which is very important. So very good. So now let me check which of the following is the correct answer, and then let us discuss about it. So the correct answer will be number C, screening plant extracts. So now these all are the way to find out new drug or discovery, but the discovery which leads to the more complex structure, unlike any other previously discussed, is the screening the plant extracts. So how this plant extracts, okay, sorry. So how this plant extract screening can be done? First is the which can be applied is a random approach. Okay, and then ethnopharmacological approach can be also done, an approach based on the traditional system of the medicine. So we all know we have various traditional home methods or traditional methods which are system of medicines which we follow in our homes, or for ancient times we have used various plant for the treatment of any kind of diseases, right. So this is the in a similar way, because the plant or the natural products are the most largest source of chemical or difficult complex structures, and these structures in the, if you have checked any natural product, you will see the nature is the one of the best chemist we can say, because in nature all the natural products, they have so much complex structure, and in one compound they are having so much chiral centers, and all the chiral centers are fixed in such a beautiful way that it affects the bodily mechanisms and any disease mechanism very particularly, and this kind of thing, making these things in the laboratory is very hard. So first is the plant selection, followed by the authentication, which can be done by taxonomic, macroscopic, microscopic, chromatographic or spectroscopic methods, and then we all also have chemometric and immunoassay DNA fingerprinting, followed by the extraction of the plant extract. And this when the plant extract is extracted, what can be what we can do is, we can do the biological and toxicological screening, okay, to check the biological activity of the plant extract, also to check if it is giving up any kind of toxic things, okay. And then what will happen, fractionation and isolation and biological screening. Then we get the pure compounds, which can be used for molecular modeling and in silico screening, followed by structural modification and new analogs. We can get the biological screening and toxicological screening, and also we can go for the structural elucidation, followed by the synthesis. Okay, so this is the way of screening, how the screening of plant extract can give us can lead to discovery of complex structure in the drug discovery and development strategies. Okay, so now I will go to the next problem. So problem number 3 says, which of the following needs to be established before the search of a lead compound takes place? The options are: number A, pharmacophore; number B, structure activity relationship; number C bioassay, and number D patents. So, which of the following needs to be established before the search of the lead compounds takes place? Already in the last to last slide I have discussed the drug discovery and development process, and also I have told you what is the steps, and in that step it is also mentioned. So, I think now all of you can, if you have followed that question very carefully, so you will be definitely able to answer this question correctly. Which of the following needs to be established before the search for the lead compound takes place? Number A pharmacophore, number B structure activity relationship, number C a bioassay, and number D patents. I am giving you some time, think, and then let me know in the chat box which of the following is the correct option. Okay, let me check the chat box. I think already all of you have answered this question very correctly. Okay. Number C, number C, number C, number B, SAR, number A. Okay, again I have lots of different answers. So, let us check and let us recapitulate what we have learned in the first slide itself. So, the correct answer will be, very good, number A bioassay. Most of you have answered it correctly. So, bioassay is the first thing which we have to establish before searching a lead compound takes place. So, now let us understand what is a bioassay. So, basically a bioassay, the basic principle of bioassay is to compare the test substance with the international standard preparation of the same and to find out how much the test substance is required to produce the same biological effect as produced by the standard. So, what are the applications of this bioassay? So, according to several pharmacopoeias, assay is used to establish the potency of a natural drug for which the chemical method is not known or established. Then standardization of drugs and natural origin like plant and animal origin whose structure and origin is unpredictable. Screening of a new compound for biological activity. Estimation of biologically active substance like histamine, acetylcholine, 5-hydroxytryptamine, adrenaline, bradykinin, substance P, prostaglandin, etc. And estimation of ED50 or TD50 divided by TD50, which is the therapeutic index, and the LD50, which presently instead of LD50 noadal LD10 is preferred. So, basically in what happens in case of the bioassay, so we can say it is basically a technique, okay, and there are several bioassay, and this bioassay are different for a different disease. So, in many disease there is, if you will see, there are several assays. Some assays are common in case of all diseases, like for an example I will give you is one is called the MTT assay. So, what happens in that particular assay, it checks if a particular substance is inhibiting the growth of a particular cells. Okay. Now, like, and for anti-cancer drug, what we check, that we have this cancerous cells in a particular tube, in a particular dish. There are several space, or there were divisions in a plate, and all the divisions we have different kind of your cells, okay, and almost equal amount of equal density of cells are given, and then we add different concentration of the particular compound which is the potent, or which is said to be the new anti-cancer compound or any kind of drug, and then we have to check if the compound is inhibiting the growth of the cells, and after we have to check it in the microscope. And this is only one example. We have different kind of assays for different disease. Sometimes when we are searching for a new compound of a drug, we have to establish an assay, and also we have to after some time we have to just recheck if our assay is potent enough by various other methods. So, again I will just give you a brief about the application of bioassay method. So, standardization of drugs of natural origin. Okay, estimation of biologically active substance like acetylcholine, adrenaline, noradrenaline, serotonin in body fluid or tissue extract. Screening of a new compounds for biological activity. Even synthetic products are subjected to these methods, as chemical substance does not truly predict the pharmacological activity. Bioassay is employed if a drug is composed of a complex mixture of substance of varying structure and activity. For example, digitalis, posterior pituitary hormones. Diagnosis and research. The concentration of gonadotropins in the blood and urine may be estimated by injecting these fluids into the animal. Like chemical methods may be employed if available. An estimation of dose of a drug is required to produce therapeutic or toxic response like ED50 and LD50. Okay. So, all these things can be done with the help of the bioassays. So, now next problem. Problem number 4 says, which of the following is not classed as an in vivo test? Number A, testing anti-cancer drug on a tumor growing on the back of a rat. Number B, testing antibacterials by measuring the growth of microorganism on petri dish of their presence. Number C, testing analgesic by determining guinea pig sensitivity to pain. And number D, testing antibacterial agents in infected rats. So, which of the following is not classified as an in vivo test? Again, I will request all of you to answer the question. After your try, I will show you what is the correct answer, and we will discuss about in detail about what is the in vivo testing. Read the options carefully. I think after that you can answer the question very easily. Which of the following is not classed as an in vivo test? Number A, testing anti-cancer drug on a tumor growing on the back of a rat. Number B, testing antibacterials by measuring the growth of microorganisms on a petri dish in their presence. Number C, testing analgesics by determining a guinea pig sensitivity to pain. And number D, testing antibacterial agents in infected rats. Let me now check the chat box. Okay, I will give you one more minute and then I will check the chat box. sorry. I think let me check, and if there are any answers, and then we can discuss it. Okay, I think I have many answers, number B, number B, number B, number A, number C. Again I have different answer, but most of you are thinking it is number B. So now let me show you what is the correct answer, and then let us discuss what is a in vivo experiment. Okay, so the correct answer will be yes, number B. Testing antibacterials by measuring the growth of a microorganism on a petri dish in their presence is not an in vivo testing. So now let us discuss about different terms we have learned so far in the medicinal chemistry. So the terms which we have heard frequently is the in silico, in vitro and in vivo. So what are these terms mean? So basically in silico it is means that performed in a virtual setting like a computer or a virtual simulation. So in silico means the computational techniques which we are using in a particular medicinal chemistry program. Okay, like molecular docking, molecular docking simulation and different screening, high throughput screening, okay, and all these things are the in silico approaches. Then we have the in vitro. So what is the in vitro method? In vitro methods are the in glass method. Okay, means the study takes place in a test tube or a petri dish or any kind of glass apparatus. It is known as the in vitro method. So in the in vitro method, what happened? We just check the growth of a particular bacteria or growth of a particular cells, cancer cells, anything. We just do it in a plate or petri dish or a particular setup, and then we test the efficacy of our compounds. But what is the in vivo? In vivo means in life, meaning the study takes place in a living organism itself. So what happens during the in vivo study? We take infected species and then we inject that particular compound directly to that particular species. Okay, so that is the in vivo. So in vitro and in vivo, these things sometimes students get confused, what are the differences and what are the similarities between the in vitro and in vivo. So what are the, let's check what is the in vitro and in vivo in details. So basically in vitro refers to a phenomenon in which a given procedure is performed in a controlled environment outside the living organism. This term is very important, in a controlled environment outside the living organism. But in vivo it refers to a phenomenon in which experiments are performed using a whole living organism. Dead organism or isolated cellular components are used in vitro, but a whole living organism is used in the in vivo. It performed under the control laboratory condition for the in vitro, but in vivo is performed under physiological condition. That is also important, because in vitro we are using the control laboratory environment, but in vivo it is happening in itself the living organism. So it is taking care of the physiological conditions of the body itself. In vitro is less expensive, but in vivo is expensive, which is quite obvious. In vitro is less time consuming but less precise, but in vivo, although it is more time consuming, it will be more precise. For example, cell culture experiment in petri dish and experiment in test tubes are examples of the in vitro, and drug testing experiments performed by model organisms such as mice, rabbit, apes, which are the in vivo. In vitro fertilization refers to artificial fertilization method in which fusion of the male and female gametes occur outside the human body. Regular fertilization mechanism in which the fusion of the male and female gametes occur within the body is referred as the in vivo fertilization. Okay. So in vivo testing system, so this is the in vitro, this is the ex vivo and in vivo. So what happened in the ex vivo, the tissues and everything are taken from a living cell, and outside that then in that living tissues taken by living cell is used for the ex vivo treatment. Okay, and what is the then difference between in vitro and ex vivo? So basically in vitro the cells are grown in a particular dish, the cells, cell culture, everything is everything is done everything is done outside the human body. For ex vivo, the cells are taken from a living body itself. So that is the difference between the in vitro and ex vivo. Sometimes these two terms can be seem to be similar, but they are stark difference in between these terms. So in vivo testing plays a vital role in drug discovery involving animal testing and clinical trials. Before a drug candidate is tested in human subject, it must undergo animal testing to further evaluate its toxicity, metabolism and over efficacy. Vertebrate animal models such as primates, dogs, rabbits are commonly used for the preclinical studies. Okay. So all of this was about the in vivo testing. So now I hope you have understood the differences between in vitro, in vivo, in silico and ex vivo testing systems, which are very important in case of drug discovery program. So now moving forward, problem number 5 says identify the correct statement about the combinatorial chemistry. Your options are: number A, one molecule can be done at one time; number B, it has high risks of failure; number C, it was developed using solution phase synthesis; and number D, it creates an impact in lead discovery and optimization. Identify the correct statement about combinatorial chemistry. First, you try to identify the correct statement. After I am showing you the answer, I will discuss in detail what is a combinatorial chemistry, which is another very important topic of today's session. Take some time. After some time I will check the chat box and then I will discuss the answer. All of you till now you have been answering every question very properly. So I want all of you again to participate and answer the question. Again, even if it is wrong, it does not matter, but answering is very important. Let me check the chat box now. Okay. So all of you you have answered very well. So number D, all of you have given answer that it is number D, which is the correct statement about the combinatorial chemistry. Now let me check if it is correct or not, and then let us discuss about combinatorial chemistry a little bit. So the correct answer will be yes, number D. It creates an impact in lead discovery and optimization. So it is the correct statement about the combinatorial chemistry. So what is the combinatorial chemistry? So the definition is combinatorial chemistry is a technique by which large numbers of different but structurally similar molecules are produced rapidly and submitted for the pharmacological assay. This technique uses the same reaction condition with the same reaction vessels to produce a large range of analogues. This technique, invented in late 1980s and early 1990s, to enable the tasks to be applied for many molecules simultaneously. What is a combinatorial library? The products produced from combinatorial synthesis are known as combinatorial library. Libraries may be collection of individual compounds or mixture of compounds. Screening the components of the library for activity using high throughput screening and techniques enables the development team to select the suitable compounds. What are the different in which steps of the combinatorial chemistry can be used in drug design? So, first is the therapeutic target, then we have the this is the flow of the drug discovery chart we have discussed earlier. First the target identification, what is the therapeutic target, then lead discovery and lead optimization, then development of candidate, and then is the drug. So, the combinatorial chemistry can make the impact during the lead discovery and the lead optimization. So, what is the difference between the conventional and combinatorial synthesis? So, in case of conventional synthesis, one molecule can be done at one time, but for the combinatorial chemistry, many molecules can be done at a time. So, conventional first is the make, then the purity, and then the test, and for combinatorial first the make, then test, and then the purity. Hundreds of molecules can be done by conventional synthesis, but for combinatorial chemistry thousands of molecules can be done in a month. Okay. The slower lead generation happens during the conventional one, but in the combinatorial one faster lead generation happens. The conventional technique has a high risk of failure, but the combinatorial technique has low risk of failure, and if used in synergy it will give us the proper lead identification in a very less short time with a high accuracy. So, this was all about the combinatorial chemistry. Okay. So, option D was the correct answer, and all of you have given the correct answer, which is very good. Okay. So, now I will move toward the next problem. So, problem number 6 is, what is the term used for the drugs that are similar in structure to a known drug and which are used for same purpose? Your options are number A, copycat drug, number B, me too drugs, number C, derivative drugs, and number D, analog drugs. The term which is used for the drugs that are similar in structure to be known to a known drug and which are used for the same purpose. What is the name of that kind of drugs? Copycat drugs, me too drugs, derivative drugs, or analog drugs. Again, take some time and let me know in the chat box A, B, C or D, which of the following you think is the correct answer. Let me check the chat box now. Okay. So, again I have many answers. Let me check for number B, number B, number C, number D. Okay, now I have three different options as answers, number B, number C and also number D I have got as answer. So now let me show you what is the correct answer and then let us discuss about it. So the drugs which have a similar structure to a known drug and are used for the same purpose are known as number B, me too drugs. So the term me too drug or follow on drug refers to a medication that is similar to a pre-existing drug, usually by making a minor modification to the prototype reflected in a slight changes in the profiles of side effects and activity and used to treat conditions which drugs already exist. Me too drugs include the diazepam, ranitidine and esketamine. The definition is a drug that is structurally very similar to a already known drug with only minor differences. The term me too carries a negative connotation. However, me too products may create competition to drive the prices down. Now, also known follow on drugs, me too drugs can be broadly defined as the chemically related to the prototype or other chemical compounds which have identical mechanism of action. Okay. So, the drug which have the similar structure of a existing drug and can be used by making a minor modification to the prototype is known as the me too drug. Okay. So, now I will go to the next problem. Problem number 7 says GNT is a given to heart patients. What is the mode of administration of this drug? Your options are number A oral, number B intravenous, number C sublingual and number D nasal. GNT is given to heart patient. What is the mode of administration of the drug? Oral, intravenous, sublingual or nasal? So, mode of administration is one other topic for the drug administration. What is the methods of administrating different drugs? So, some drug you have seen, some drugs are given as a tablet or a capsule formation. So, that is the oral way. Some drugs are given as injected formation, that is the intravenous method. Some drugs are given as sublingual. Sublingual means by the oral method, by keeping the drug inside your under your tongue. And the nasal method is the method of your throughout your nose or the nasal pathway. So, GNT is a famous drug which is given to the heart patients for which you have to identify what is the mode of the administration of this drug. Let me check the chat box now and then I will discuss about it. Okay, many of you have answered it, which is very good. Number A, number B, number C, number C. Okay, so I think I again have multiple options as the answer. So let me check which is the correct one and then let us discuss about it. So the correct answer will be number C, sublingual. Okay, so now let us check. So this is the structure of the GNT, which is the glyceryl trinitrate. Okay, this is a very famous drug for which is given to the heart patient. So some drugs can be absorbed through the mucous membrane of the mouth or nose, and they can avoid the digestive and metabolic enzymes. Especially relevant when you are looking for at a heart patient that take glyceryl trinitrate or GNT, and they place this drug under their tongue, and this is known as a sublingual administration. So advantages of the sublingual administration is it is economical, quick termination, first pass avoided, and drug absorption is quick. And the disadvantage is unpalatable and bitter drugs, irritation to the oral mucosa, large quantities cannot be given, and few drugs are absorbed in this way of the sublingual. So sublingual way of administration is keeping the drug just under the tongue, and the drug will be absorbed from this particular mucous membrane of the mouth. Okay, and this will help to this drug to avoid the digestive and metabolic enzymes, and also the first pass is avoided. Okay. So remember, GNT, which is important for heart patient, is given by the sublingual pathway. So now next question, problem number 8 says the intrathecal injection is used for number A, bacterial infection in the brain, number B, bacterial infection in the spleen, number C, fungal infection, number D, viral infection. Intrathecal injection is used for which in kind of infection? Number A, bacterial infection in the brain, number B, bacterial infection in the spleen, number C, fungal infection, and number D, viral infection. Again, I want all of you to participate. Till now, all of you are answering the questions and following the question answer session, which is very important. I will give you some more time and then I will show you the answer and we will discuss about the intrathecal injections. So, for which this kind of injection is used? Bacterial infection in the brain, bacterial infection in the spleen, fungal infections, or viral infection. Okay, now let me check the chat box. Okay, so many answers are given. Number A, number B, number A. So number A is the most given answer. Okay, let me check if it is the correct one or not, and I think I also have number B as answer. Okay, so the correct answer will be, okay, this is number A. I just have made this mistake. I will just correct it quickly. Yeah, number A is the correct answer, bacterial infection in the brain. Okay, just, yeah. So yeah, number A is the correct answer, bacterial infection in the brain. Okay. So this last form, or this was a continuation. So intrathecal injection is basically the injection into the spinal cord. Now it is a very frequently used in the situation where you have a bacterial infection in the brain, and administration of an antibacterial drug is not very useful because it does not cross the blood brain barrier. In the last class we have discussed detail about the what is a blood brain barrier, okay, and what does it do. So we have discussed it in detail in the last session. If you have not attended the last class, you can just have a look at the video which I have uploaded for the last class. There we have discussed about the blood brain barrier in very detail. So if you give this kind of intrathecal injection, you can avoid the crossing the blood brain barrier, because this can be used for administration of anti-cancer drug and anything which is for the brain. So in case of the brain, we all know there is a blood brain barrier for which many hydrophilic drugs, they cannot pass through this particular blood brain barrier, and readily hydrophobic drugs is can pass easily from this blood brain barrier. But most of the drugs which we use for different problems, they do not cross generally this blood brain barrier. So this is by intrathecal injection, means the injecting into the spinal cord, it will help the particular drug to pass through the brain itself, okay, but it do not need to avoid crossing the blood brain barrier. Okay. So it means the drug is injected into the spinal cord. Antibacterial agents do not normally cross the blood brain barrier and often administered in this way. Intrathecal injection are used to administer methotrexate in the treatment of childhood leukemia in order to prevent the relapse in the CNS. Okay. So number A was the correct answer, for which most of you have answered it correctly. So very good. So now I will show you the next problem. Problem number 9 says pores in the nephron cell membrane which are specific for reabsorption of the water molecule and prevent the passage of iron or other molecules are called, number A, glomerulus, number B, arterioles, number C, aquaporins, and number D, none of these. So what are the pores of the nephron cell membrane which are specific for the reabsorption of the water molecule and prevent the passage of the ions or other molecules are called? Glomerulus, arterioles, aquaporins, or none of this. If your answer is number D, none of this, then let me know in the chat box what are these pores called. Again I am repeating, pores in the nephron cell membrane which are specific for reabsorption of the water molecules and prevent the passage of the ions and other molecules are called glomerulus, arterioles, aquaporins, or none of these. If your answer is none of this, then I want to know what are these pores called. So in the chat box, I want all of you to participate again and answer A, B, C or D is the correct answer. If your answer is D, then let me know what the name of the pores in the nephron cell membrane which are specific for reabsorption of water molecule and prevent the passage of iron and other molecules. I am giving you some time again. Think and then let me know. let me check the chat box now. Okay, so I have many answers, I think. Number A, number C, number A, number C, number A, number C. So you are divided into the two part, either it is number A or number C. Okay, so let me check if number A or if number C is correct, and then let us discuss it. So the correct answer is number C, aquaporins. So aquaporins are the pores in nephron cell membranes which is specific for the reabsorption of the water molecules and prevent the passage of the ions or other molecules are called. Okay. So this is because nephron is surrounded by a rich network of blood vessels carrying the filtered blood away from the glomerulus, permitting much of the contents of nephron to be reabsorbed into the blood supply. Most of the water that was filtered into the nephron is quickly reabsorbed to the pores of nephron cell membrane, which are specific for water molecules and bar the passage of ions and other molecules. These pores are made of the protein molecules which are known as aquaporins. So aquaporins are also called the water channels, are integral membrane proteins from a large family of major intrinsic protein that form pores in the membrane of biological cell, mainly facilitating the transport of water between cell. The cell membranes of a variety of different bacteria, fungi, animal, plant cells contain aquaporins through which water can flow more rapidly into or out of the cell than by diffusing through the phospholipid bilayer. 6 membranes spanning alpha helical domain which both carboxylic and amino terminal on the cycloplasmic side, 2 hydrophobic loops contain concerted asparagine, proline, alanine motif. These aquaporins selectively conduct the water molecules in and out of the cell while preventing the passage of iron and other solutes. Aquaporins are integral membrane proteins. Okay. So the correct answer were aquaporin. So now next question, which is a mathematical question. So a patient received a single intravenous dose of 460 milligram of a drug substance that is produced an immediate blood concentration of 9.5 milligram per liter. Now calculate the apparent volume of distribution. Your options are number A 0.027 per liter, number B 0.0211 per liter, number C 47.4 liter and number D 48.4 liter. Again I will repeat, a patient received a single intravenous dose of 460 milligram of drug substance that is produced and that produced an immediate blood concentration of 9.5 milligram per liter. Now calculate the apparent volume of distribution. I will give you 2 minutes, try to answer this question, then after I will show you what is the volume and distribution and how to solve this kind of problem. But first all of you try to solve this problem, it is very easy, just try to solve it. If you know what is the volume of distribution, then if you have read about it, you will be able to answer the question more easily. I am giving you 2 minutes, try, and then I will show you the answer and we will understand or discuss what is the volume of distribution. Okay, let me check the chat box and then I can show you how to do this kind of mathematical problem after checking the answers. Okay, I think I have few answers by now. Number A, number C. Okay, number A is the most given answer. Okay, let me check if the number A is the correct one. So, the correct answer will be number D, 48.4. So, first let us understand what is the volume of distribution. So, the apparent volume of distribution or VD measures how well a drug is distributed outside the vascular space, and it is defined as the VD of a drug represents the degree to which a drug is distributed in body tissue rather than the plasma. VD is directly proportional with the amount of drug distributed into the tissue. A higher volume of distribution indicates a greater amount of tissue distribution. In rough terms, drugs with a high lipid solubility or low rates of ionization or low plasma protein binding capabilities have higher volume of distribution than drugs which are more polar, more highly ionized or exhibit high plasma protein binding in the body's environment. The formula of volume of distribution, which can be given in ml if it is in ml. So, the amount of the drug in the body, okay, in microgram or blood or plasma concentration. The units are all relative, okay, whatever. So, the volume of distribution equal to amount in the body divided by the blood or plasma concentration. So, if now you see the question, the amount of drug is given 460 milligram, right, and the immediate blood concentration is 9.5 milligram per liter. So, now what will be the how to solve the drug? So, the volume of distribution will be 460 milligram divided by 9.5 milligram per liter, which will give us 48.4 liter. So, this is the apparent volume of distribution. Okay. So, now I will move toward the next problem. So, problem number 11 says, what is the term used for an animal that has been genetically modified for in vivo test? Your options are number A, chimeric animal, number B, transformed animal, number C, hybrid animal, and number D, transgenic animal. What is the term used for an animal that has been genetically modified for in vivo test? Is it chimeric animal or transformed animal or hybrid animal or transgenic animal? Let me check the chat box. Okay, so there are many answers again. So, number D is the most given answer, or the only given answer is the number D. So, now let me check if number D is the correct one or not. So, the correct answer is the, yes, number D, transgenic animal. So, what is the definition? An individual in which a gene or genes were introduced by one or other technique of transfection to the genome of an animal is known as transgenic animal. Or a transgenic animal is one that carries a foreign gene and has been deliberately inserted into its genome. The gene introduced by the transfection is called the transgene, and the transgenesis may be defined as the introduction of exogenous DNA into the genome, such as the stably maintained in the heritable manner. So, what is the difference between the chimeric and transgenic organisms? For chimeric organism, it is a single organism composed of cells which are more than one distinct genotype, and for transgenic organism, it is the organism created combining the DNA from different genomes or insertion of a foreign DNA into the genome or other organism to another. The process a result of combining the cells two or more genetically distinct individual. The transgenic organism a result of combining foreign DNA into an organism. The transgenic chimeric organism does not always bring out the changes to the phenotype of the individual. Transgenic organism always brings out the changes to the phenotype of individual. The new individual have the cells with more than one distinct genotype is the chimeric organism, and for in case of transgenic one, new individuals have transgene. For example, chimeric mice is the example of chimeric organism. Transgenic plants, animals and microorganisms are the example of transgenic organisms. Okay. So, the correct answer was transgenic animal. So, now I will move toward the next problem. So, problem number 12 says, which of the following statement is false regarding the retrovirus? Your options are: number A, they contain RNA as genetic material; number B, reverse transcriptase enzyme is present in retrovirus; number C, proceeds through the DNA independent steps, okay, and number D, none of the above. So, which of the following statement is false regarding the retrovirus? Number A, they contain RNA as a genetic material. Number B, reverse transcriptase enzyme is present. Number C, proceeds through the DNA independent steps, and number D, none of the above. Again, I will give you 2 minutes. All of you try to answer the question. After that, I will show you the answer and we will discuss about retrovirus. So, whenever this kind of true false or correct incorrect questions are given, please be careful, because most of the students make mistakes in this kind of questions. First read the options carefully and then identify which will be the correct answer. Now, let me check the chat box. Okay, so the answers are given, number D, number C, number D, number A. So, now I have multiple answers for this question. So, let me check which of the following is the correct one and then let us discuss about it. So, the correct answer will be number C, which is the, okay, so I think somebody wants to tell me anything. Okay, so if you have to say anything, you can just write it in the chat box. Okay, I will just answer your query after I discuss this question. I will answer your query. So Statement number C was incorrect regarding the retrovirus. So now let us check what is the structure of the, what is the morphology of retrovirus. So basically you can see the outer protein core is known as the, it is the envelope glycoprotein. After that we have a lipid envelope, which is this one, okay, and this is the envelope glycoprotein, this is the lipid envelope. Then inside we have this particular different enzymes. First this is the matrix, okay, this one is the matrix, and then we have different enzymes like protease, RNA matrix, okay, then we have the RNA, this is the RNA, this is the protease, then matrix, this is the matrix, this is the code, okay, and there is the reverse transcriptase enzyme, then we have integrase enzyme and nucleocapsid. So interesting points about retroviruses are this: retrovirus contain RNA as genetic material but have DNA dependent step in their replication. So this is the DNA independent step, this is the replication, it does not happen. They, although their genetic material is RNA, they have DNA dependent steps in their replication. Replicates via reverse transcription because the presence of reverse transcriptase enzyme. Integrase transfers the viral DNA into the cell nucleus, and viral double stranded DNA is covalently randomly integrated into cell's genome. Retrovirus that can transform host cell at high rates contain gene sequence such as viral oncogenes and proto-oncogenes. Human retrovirus can cause immunodeficiency, cancer and neurological disease. Okay. So statement number C was the false regarding the retrovirus. So now I will move forward. So problem number 13, it says that which of the following is false regarding the renin-angiotensin system. Your options are: number A, aldosterone is a steroidal hormone; number B, vasoconstriction is caused by the action of angiotensin 1; number C, aldosterone enhances the retention of the sodium and water in the blood; number D, bradykinin is destroyed by ACE or angiotensin converting enzyme. Which of the following is false regarding the renin-angiotensin system? Again I will give you 2 minutes, just try to answer the question. Even if your answer is wrong, it does not matter, I am telling you. Just try to answer the question. Which of the following is false regarding the renin-angiotensin system? Number A, aldosterone is a steroidal hormone; number B, vasoconstriction is caused by the action of angiotensin 1; number C, aldosterone enhances the retention of sodium and water in the blood; and number D, bradykinin is destroyed by the ACE or angiotensin converting enzyme. So let me check the chat box. Yes, I have many answers already given. Number B, number C, number D, number B. Okay, so again I have different answer for this question. Let me show you the answer and let us discuss about it. So the correct answer will be number B. Vasoconstriction is caused by the action of angiotensin 1. This is the false statement. So this is the renin angiotensin aldosterone pathway. So first, what is the step is the dehydration or sodium deficiency or hemorrhage, which can lead to the decrease in the blood volume and decrease in the blood pressure. So if there is a decrease in the blood pressure, what will happen? Juxtaglomerular cells of kidneys are triggers which will increase the flow of the renin. Okay, and then in the from the liver, angiotensinogen is also secreted, which will together will increase the amount of angiotensin what, which is converted to the angiotensin 2 by the work of the ACE or angiotensin converting enzyme. Now this angiotensin 2, it will be doing few things, like first it will lead to the vasoconstriction of arterioles, which will increase the blood pressure. Okay, and also it will again it will just stimulate the adrenal cortex, and which is also for the increased potassium and extracellular fluid, which will increase the aldosterone. This aldosterone in kidneys to increase the sodium and water reabsorption, which together increases the blood volume. Okay, the vasoconstriction of arteriole and the increased blood volume, both of this will increase the blood pressure until it gets to the normal. So the vasoconstriction, it is not caused by the action of angiotensin 1, but it is caused by the action of angiotensin 2, which is converted from angiotensin 1 by the act of the enzyme ACE or angiotensin converting enzyme. So this is the other angiotensin in case of aldosterone, which is different aldosterone is going to for various wave in the sodium retention and high blood pressure. Okay, like the reason of high blood pressure are various, like arterial stiffening or arteriosclerosis, renal fibrosis, pituitary chlorine renal function, cerebrovascular disease, stroke or fibrosis, and etc. Okay, so this is the renin, angiotensin and aldosterone pathway, which is very important in maintaining the blood pressure in our body. So now I will just move forward to the next problem. So problem number 14 says beta blockers are used as dash agents. Your options are number A anti-diabetic, number B anti-hypertensive, number C antibacterial, and number D antifungal. Beta blockers are used as dash agents: anti-diabetic, anti-hypertensive, antibacterial or antifungal. Beta blockers are which kind of agents? Anti-diabetic, anti-hypertensive, antibacterial or antifungal? I will give you one more minute and then I will check and we will discuss this. Let me check the chat box. Okay, number B is the given answer. Let me check if number B is correct or not. So the correct answer is yes, it is number B, antihypertensive. So there are antihypertensives are used for the higher blood pressure. Okay, for the this is the blood pressure medicine. So there are different classes of antihypertensive drugs. So, let us discuss few of them. There is diuretics. Okay, first class is the diuretics. The mechanism of action of diuretics is it acts on kidneys to increase the excretion of sodium and water, decrease the blood volume and decrease the blood pressure. For example, there are thiazides like hydrochlorothiazide, chlorothianol, high ceiling like furosemide, cleptospirine like spironolactone, etc. We have angiotensin converting enzyme inhibitor, which is a ACE inhibitor, which inhibits the transformation of angiotensin 1 to angiotensin 2. So, this is the mechanism of action which inhibit the synthesis of angiotensin 2 and decrease the peripheral resistance and blood volume. For example, captopril, linsopril, enalapril. So, these all are the ACE inhibitor. Then we have angiotensin 81 blockers like losartan, valsartan, telmisartan. So, these are the 81 blockers. So, these are mechanism of action is blocks the binding of angiotensin 2 to its receptor. And then we have adrenergic receptor antagonists which are known as the beta blockers. For example, propranolol, nebivolol, atenolol. The mechanism of action is competitive antagonists that block the receptor sites on adrenergic beta receptors. Okay, so these are the different classes of antihypertensive drugs with their mechanism of action and the examples. Okay. So now moving forward, the problem number 15 says the usual amount of drug in a tablet is approximately -%. Your options are number A 20 to 25, number B 5 to 10, number C 1 to 5 and number D 15 to 20. So, what is the usual amount of drug in a tablet? Number A 20 to 25, number B 5 to 10, number C 1 to 5 and number D 15 to 20. Identify the correct option and let me know in the chat box. The usual amount of drug in a tablet is approximately -%. Let me check the chat box if you have given the answer or not. Okay. Number A, number B, number D. So, I think I have different answer for this question. Okay. I will discuss it now. Okay. Since we don't have much time left for today's class. Okay. So, the usual amount of drug in a tablet is approximately number B 5 to 10%. So, now tablet is a compressed preparation that contains maybe 5 to 10% of the drug and about 80% of other fillers, and there are some disintegrant, lubricant, gradients and binders. So, on about 10% of compounds to ensure easy disintegration, disaggregation and dissolution of a tablet into the stomach or the intestine. This is a process which is defined as the pharmaceutical phase of the drug action. Of course, we know, we will not be spending much time on that part. Okay, so the entire industry that revolves around how to deliver the drug to administer at every efficient manner. So, there are pills that can be coated with sugar, varnish or wax to help to disguise the taste, because some of the drugs are extremely bad tasting that helps us to ensure that drug is properly consumed. Many of us have taken drugs as capsule. A capsule is nothing but a gelatinous envelope which encloses the active substance. Okay, and this capsule can be designed to remain intact for some hours after the ingestion in order to delay the absorption. So, the usual amount of drug in a tablet is approximately 5 to 10%. So, now next problem. Problem number 16 says which of the following is the correct definition of bioavailability. Number A, bioavailability describes the proportion of the drug administered that is metabolized very quickly, that is not available to induce a physiological effect. Number B, bioavailability describes the ability of an administered drug metabolized to cause the undesirable physiological effect. Number C, bioavailability is used to describe the fraction of the drug of a dose of the drug administered that is present within the body and facilitates the desired physiological effect. And number D, bioavailability is the length of the time an administered drug is present in the body that is available to cause a physiological effect. So, what is the correct definition of bioavailability? Read each and every option very carefully and then let me know in the chat box A, B, C or D is the correct definition of bioavailability. Again I will give you 2 minutes, try to answer it within 2 minutes, okay. Then I will show you and we will discuss about it a little bit. What is the correct definition of bioavailability? I will give you one more minute and then I will check and we will discuss about it. Okay, let me check the chat box now and then we have to discuss about it. Okay, so we have number C. Okay, so number C is the given answer for the definition of bioavailability. Now let me check if number C is the correct one or not. So yes, bioavailability is used to describe the fraction of a drug dose of a drug administered that is the present within the body and facilitates the desired physiological effect. So bioavailability is the fraction of administered drug that reaches to the systematic circulation. It is expressed as the fraction of the administered drug that gains access to the systematic circulation in a chemically unchanged form. For example, if 100 mg of a drug is administered orally and 70 mg of this drug is absorbed unchanged, the bioavailability is 0.7 or 70%. The definition is the, again I am have repeated it here. So this is the significance. What is significance? Dosage of the drug of higher hepatic extraction. Okay, the drug in tablet, first it is we have the dissolved drug, then we have which the components in stomach and intestine, then we have the in the gut wall we have the absorbed drug, and in the then after that the drug in the portal vein. Okay, this is the bioavailability of a drug which is given as the term F, which is denotes the bioavailability. Okay. Now next problem, when a medicine is added intravenously, its bioavailability is number A 50%, number B 25%, number C 100% and number D 98%. So what is the bioavailability of a medicine which is given intravenously? What is the bioavailability of a medicine which is given intravenously? Let me check the chat box. Okay. So, number A, number D, number C. So, again I have multiple answers for this question again. Okay. Let me check which of the following is the correct one. So, the correct answer will be number C, 100%. So, now in what I have mentioned earlier also, in pharmacology, bioavailability BA or F is a subcategory of absorption and is the fraction or percentage of an administered drug that reaches the systematic circulation. By definition, when a medication is administered intravenously, its bioavailability is 100%. Okay, bioavailability, fraction of absorbed into the systematic circulation of the drug's bioavailability. Okay, this is the GI lumen and this is the gut wall. Okay, and this is the portal vein, this is the liver, and this is the systematic circulation. Okay, so bioavailability, the formula bioavailability is the area under curve of this drug and the oral by the area of the under curve in the intravenously into 100%. Okay, and bioequivalence is the term in pharmacokinetics used to access the expected in vivo biological equivalence of true proprietary proportion of drug. If two products are said to be bioequivalent, means they would have expected to have all intended person is the same. Okay, for so this is the if the fraction value is 100%, if the systematic, it is given directly to the systematic circulation. Okay, so the correct answer was 100%. So now next problem. So this is a mathematical problem. Okay, I want all of you to try to answer this question. If the bioavailability of a drug of digoxin is 0.55 milligram tablet is 0.40 compared to the bioavailability of 0.80 of digoxin elixir, which is the concentration is 0.07 milligram per ml. Calculate the dose of the elixir equivalent to the tablet. Again I will repeat, if the bioavailability of this drug in a 0.55 milligram tablet is 0.40 compared to the bioavailability of 0.80 in a elixir, which the concentration is 0.07 milligram per ml. Calculate the dose of the elixir which is equivalent to the tablet. Your options are number A 3.928 ml, number B 8.758 ml, number C 3.339 ml and number D 5.006 ml. I will give you a minute or two. Try to answer the question. Is very easy. Just read the question carefully. Then you can easily answer the question. If the bioavailability is 0.40 in a 0.55 milligram of tablet and the bioavailability of is 0.80 in a elixir for which the concentration is 0.07 milligram per ml, calculate the dose of the elixir which will be equivalent to the tablet. Let me check the chat box now. I think. Okay, I will give you one more minute. Okay, let me check if you have answered it or not. Then I will show you how to solve this kind of problem. Okay, I have two answers, number D and number C. So two students have solved it till now. Let me show you the answer. Then you will understand how to solve this kind of problem. So correct answer will be number A, 3.928 ml is the correct answer. So see, The question says that the drug bioavailable from a tablet is 0.40 from a 0.55 milligram of tablet. So, what is the amount which is bioavailable? That is 0.05, that is 40% of this 0.55 mg tablet means 0.22 mg. Now, same bioavailable the drug which is bioavailable from the from per ml of elixir is 0.05. In 1 ml of elixir we have 0.07 milligram of that particular drug, and the bioavailability is 0.80 means 0.056 mg is bioavailable from 1 ml of elixir. So, now we have to calculate what is the ml of the elixir which have this 0.22 milligram of the particular drug. So, 0.056 milligram is available in a 1 ml of elixir. So, means 0.022 milligram is available from 3.928 ml of that particular elixir. Again I will repeat, the drug which is bioavailable from the 0.55 milligram of tablet, the bioavailability is 0.40 means what is the how much mg of the drug is bioavailable is 0.55 into 0.40 which is 0.22. So, now the bioavailability which is in the per ml of elixir is, in 1 ml we have 0.07 milligram of the drug and the bioavailability is 0.80 means 0.056 mg. Now, the question was how the calculate the dose of elixir equivalent to the tablet. So, from one tablet we are getting 0.22 milligram, and for 1 ml of elixir we are having 0.056 milligram. So, the ml which is equivalent to the 0.55 mg of tablet is this. Okay. So, that is the correct answer, number A. So, now next question, it is also another mathematical question. It says if area under curve of an oral dose of a drug administered is 6.75 mg per ml per hour and the intravenous dose is 13.5 mg per ml per hour, calculate the bioavailability of the oral dose of the drug. Number A 30%, number B 40%, number C 15% and number D 50%. I have mentioned it when I was discussing the bioavailability, how to calculate the percentage from the area under of curve. Okay. So, now try to answer this question. If area under curve or AUC of an oral dose of a drug is 6.75 and the intravenous dose is 13.5, what is the bioavailability? Let me check the chat box now. Okay. Number A and number D is the most given answer. Let me check if which one is correct. So, the correct answer is number D, 50%. So, how to calculate? Again I have discussed about the bioavailability. Okay. So, it is when a systematic availability of a drug administered orally is determined in composition to its intravenous administration. Okay. So, there are different standards are used, like intravenous dose is selected as standard, intramuscular dose is for the poorly water soluble drug, and oral solution is used for certain case. So, the bioavailability of a drug may be determined by comparison of AUC data for the particular dosage of the against the intravenous form. So, the bioavailability, the formula is AUC in a dosage form divided by AUC by intravenous. So, AUC or intravenous is the your standard. So, in the question it is given that the oral formation it is 6.75 and mg per ml per hour, and in the doses it is the 13.5 mg per ml per hour. So, the answer will be 6.75 divided by 13.5, which is 0.5 or 50%. Okay. So, 50% was the correct answer. So, now last question of today's session, it says that the following diagram represents the phenomena which is known as SPR or surface plasmon resonance. What is represented by D? Here D, what is represented by this D? Number A, monochromatic plane polarized light, number B, flow of the buffer solution, number C, receptor, and number D, evanescent wave. In this particular graph which in this particular diagram which is representing the phenomena of SPR or surface plasmon resonance, what is represented by D? Monochromatic plane polarized light, flow of the buffer solution, receptor, or evanescent wave. So, I am giving you a minute, try to answer the question, then I will show you the answer, and we will this is the last question of today's session. Let me check the chat box because I have to discuss it again. Okay. The answer is given is number D, and number A is the given answer. Now, let me check and let us discuss the last question of today's session. So, the correct answer will be number D, evanescent wave. So, basically this number C is the flow of the buffer solution, number D is the evanescent wave, number A is the immobilized ligand, number B is the plane polarized light, and this is the detector and the glass plate. So, what is it all about? The surface plasmon resonance is the optical method of detecting when a ligand binds to a target. The procedure is patented by Pharmacia Biosensor as Biocore, and it makes the dextron coated gold sulfate glass chip. A ligand that is known to bind a target is immobilized by linking it covalently to the dextron matrix, which is in the flow of the buffer solution. Monochromatic or plane polarized light is shown to the angle of alpha angle of incidence alpha from below of the glass plate and reflected back at the interface between the dense gold coated glass and the less dense buffer solution. However, a component of a light is known as effervescent wave penetrates a distance of about one wavelength into the buffer and dextran matrix. Normally, all of the light including the effervescent wave is reflected back, and if the gold flame is very thin fraction of the effervescent wavelength and the angle of incidence is exactly right, the effervescent wave interacts with the free oscillating electrons known as plasmons in the metal flame. Okay. So, the number D was the correct answer. So, this was the last question of today's session. So basically what we discussed today, we discussed about the lead discovery. First we started with lead discovery, then we known various terms about the drug discovery processes, then we moved toward the bioavailability and we discussed few mathematical questions regarding bioavailability. I will share this lecture PPT and this video with you shortly. Please check, and if you have any doubt or any query, feel free to reach me through the email. I am sharing my email ID here in the chat box. Okay. So, if you have any doubt or any query, just send me an email. In this email ID, I will feel happy to answer your queries. So, today it was our week 6 live session. I thank all of you to join and answer. The question answer session was very good today because all of you have participated in very well. So, thank you all for joining and participating in the question answer session. This kind of live doubt clearing session we will be having in every week from on Tuesday from 6 to 8 p.m. from this on this particular Google Meet ID. So now I will close the session for today. So thank you all for joining. If you have any problem, any doubt, feel free to reach through this email ID. Okay, and I will close the session for today. This video and lecture PPT I will be sharing to you in the SWAYAM website shortly. So thank you all. Let's meet on the next week on next Tuesday. And if you have any problem, please feel free to reach me through the email ID. Okay, thank you. I will close the session for today. Thank you all.